Today we are going to discuss the mediators of inflammation, the chemical mediators. The chemical mediators could be cellular mediators or plasma mediators. Now what is the difference? Cellular mediators means they are synthesized, they are produced by cells like macrophages. Majorly produced by macrophages, but there are other cells also like dendritic cells, mast cells, then platelets or neutrophils. Now they can be either preformed or they can be synthesized de novo. Now preformed mediators are like histamine, serotonin, lysosomal enzymes, gelatinases or acid hydrolases. Or synthesized de novo could be nitric oxide, arachidonic acid metabolites like prostaglandin leukotrienes or platelet activating factors or cytokines or chemokines. Now, plasma mediators means they are synthesized mainly in the liver as inactive precursor and they are activated by the proteolytic cleavages. Now, these include the complement system, the Kinnin system, and the clotting system. Now, going with the cellular mediators first, the preformed cellular mediators. Histamine. Histamine is formed from an amino acid. Histamine is formed from an amino acid histidine. It is formed from an amino acid histidine and the sources are mast cell majorly and also platelets and basophils. Now what does it causes? Histamine we can remember from allergic reactions or anaphylaxis. Therefore, it causes vasodilation, increased vascular permeability and bronchospasm. So histamine causes all of these things. Serotonin, 5-HT, the richest source is platelet, but it is also present in enterochromaffin cells. The main function is it acts as a neurotransmitter in GIT and the actions are similar as that of histamine, that is vasodilation, increased vascular permeability and bronchoconstrictions. Now the third one is lysosomal enzymes. They are found in the lysosomes of the neutrophils or monocytes and the role is intracellular killing of microbes or dead cells. And then we have gelatinases and acid hydrolases. What is the source? Source is tertiary granules or C particles of the neutrophil. Okay, these are also involved in intracellular killing. So these are the preformed mediators: histamine, serotonin, lysosomal enzymes, gelatinases, and acid hydrolases. Now coming to newly synthesized cellular mediators like nitric oxide. L-arginine, how it is formed? L-arginine, in the presence of the enzyme nitric oxide synthase, converts it into nitric oxide, and the function is in intracellular killing. It is also known as endothelium derived relaxing factor. It causes relaxation of the vascular smooth muscle, therefore it acts as a vasodilator. Also it decreases the platelet aggregation and it is a direct micropecidal action also. Then arachidonic acid metabolite, we can see this diagram. This is the membrane phospholipid. In the presence of the enzyme phospholipase A2, it converts into arachidonic acid. Now whether cyclooxygenase enzyme or lipoxygenase enzyme acts on it, they are different mediators. Now with the cyclooxygenase enzyme, it leads to prostaglandin formation, PGG2 then PGH2. From PGH2, it can form either prostacyclin or thromboxanes or the prostaglandins. Now, prostacyclins and thromboxanes have opposite actions. Prostacyclin, they act as vasodilators, while thromboxane act as vasoconstrictors. Prostacyclin, it inhibits the platelet aggregation, while the thromboxane, it stimulates the platelet aggregation. While prostaglandin PGD2 and PGE2, it causes vasodilation and it increases the vascular permeability. Now, talking about the lipoxygenase pathway, it leads to the formation of 5-HPETE. What is HPT? Hydroperoxy eicosA tetraenoic acid. Now this leads to formation of leukotriene A4. Now leukotriene A4 to leukotriene B4 or C4D4E4. B4 is act as a chemotaxis agent. Also from HPETE to HETE. HETE also act as a chemotaxis agent. Now LTC4, D4 and E4, it causes vasoconstriction, bronchospasm and increased vascular permeability. If you remember, these are known as slow reacting substances of anaphylaxis. Okay. There is one more pathway from HPET in presence of 12 lipoxygenase enzyme. It leads to lipoxin A4 or lipoxin B4. These lipoxin A4 and B4, how does they act? They inhibit the neutrophil adhesion and they inhibit the chemotaxis. Okay, so these are the second. I am talking about the newly synthesized cellular mediator. We have talked about nitric oxide, then arachidonic acid metabolite. Now talking about cytokines. Cytokines are produced by activated lymphocyte, macrophages and dendritic cells. Also, they are produced by endothelial cells, epithelial cells and connective tissue cells, and they mediate and regulate immune and inflammatory reactions. Now, there are three important features we study about cytokines, that they are pleiotropic, they are redundant, and they have a cascade effect. Now, what does that mean? Pleiotropy means one cytokine can have different effect on different cells. Redundant means different cytokines can have the same effect. And cascade means cytokine can stimulate the production of other cytokines.